Okay. This PowerPoint is it is on Moodle, so if you want to see other parts of it, then it's easily, if you want to come back to it other than what you've got in your handouts, then it is all there. This idea of mutation, then, it's a word that's sort of bandied about in a very general sort of way, isn't it? And at this level, it actually has some very specific meanings, and there are some very specific types of mutation that change the DNA code. Now, if you change the DNA code, you change the sequence of amino acids that are produced from it, probably, and consequently you change the protein that you get. It might change it so that it doesn't work at all. It might just not form. It might actually produce a better version. And of course, you've got to remember that if you you fully subscribe to the evolution of life on Earth through biochemical change, then we are simply mutated bacteria. The first cells on the Earth were bacterial cells, the prokaryotes. So we seem to have come quite a long way through mutation and then selection of mutations. And we finished the last unit with a bit on evolution. Not a lot, but it does all rely on variation in populations, which really comes about through mutations. It's the only way you can get changes in the genetic code, really, is mutation, isn't it? You've got to change it somewhere along the line. So mutations are not all bad. They can provide the raw material for evolution and can be definitely an advantage. The idea then of two different types of mutation is really just a matter of scale more than anything else. So chromosomal mutation is one affecting the whole of a chromosome or part of a chromosome. You can get multiple copies, you can get chunks of chromosomes cut off one and transferred to another, you can get bits exchanged. So those are chromosomal. And they will have, or they will tend to have, much broader impacts on individuals because of the number of genes on a chromosome. I mean, we're averaging about 1,000 genes per chromosome. I mean, there aren't 1,000 genes per chromosome because they differ so much in size. So you'll have some that are down in the few hundreds and others that are several thousand. But, if you have an extra copy of a whole chromosome or you're missing a whole chromosome, then the impact is obviously through all of those genes. So it's a much larger scale. Gene mutation, on the other hand, is changing the base sequence in a single gene. We presumably get mutations also between the genes in the non-coding DNA. And we probably thought in the past that that didn't matter. But we now know that actually it does. And there are certain conditions which are related to getting certain numbers of repeats in the interfering bits of DNA, the introns. And it can affect the expression of a gene quite considerably. So we'll look at a variety of different mutations to start with. First of all, you've got to consider how they come about. Well, there are two points at which mutations commonly occur. On the left, if you get a mutation in what we'd call germ cells. Now, germ cells are reproductive cells. So in humans, we're talking egg cells and sperm cells. So at that point, if you've got a mutation right at the beginning of life, either in a sperm cell, in an egg cell, or in the newly formed fertilized egg, what we call a zygote, then every cell from there on is going to be affected because that mutation is then copied and passed on, copied and passed on, copied and passed on. So, I mean, the mutation might have occurred before that, but it can occur at that stage. And so on the left, we've got the impression is supposed to be that the mutation is in red, and so the entire organism carries that mutation. And it's much more easy to pass on. The other option on the right-hand side there, you've got somatic mutation. Now, somatic, the impression given there is that it's a mutation that occurs in an embryo. It doesn't have to be an embryo. It could happen, in a fully grown individual. You can get a mutation occurring, but only cells that arise from the one that's been mutated will carry the mutation. So the impression given there is that, some effect has caused a mutation in a patch of cells which go on to become the left shoulder. And it's, you know, it is, again, a matter of scale. So mutations early on will affect more cells than mutations later in life. Now, if we look at different types of gene mutation, what we have are, well, the causes can be various. And I've got a odd sort of selection there. I think probably the top one is going to be the most common, what we just described as a biochemical accident. If we're copying the DNA when cells divide, there's huge scope for errors. I've forgotten what it said on that website now. Is it 3.5 billion bases or something in our genome? You know, we're talking billions of bases. Now, when a cell divides, you've got to make an exact copy of that to pass on to each of the new cells. So there is a huge amount of scope for error, isn't there? I mean, you think how easy it is if you're copying something off the board to make a spelling mistake or miss a word out, or, you know, write the wrong word, or, you know, sometimes repeat a letter. Easily done, isn't it? And you can just cross it out and rewrite it. But of course, you know, genes are not like that. So there is a certain degree of proofreading available in the DNA, which is remarkable. You know, we actually have enzymes that check when DNA is being copied, that it's been copied accurately. It's sort of comparing the old copy and the new copy. Do they match up? And if not, then they take the new copy apart and it's rebuilt. So we do have proofreading, but it's not totally foolproof. You know, it doesn't always work. If it did, then you wouldn't get any mutations passed on through that mechanism, and we quite clearly do. And that can happen, well, that's the failure in the DNA repair process that I've put up there. So, you know, it could just be copied wrong or failed repair. And the scope, you know, particularly noticeable, I guess, very early in our lives, you know, and I'm talking very, very early in our lives, in, you know, first few days and weeks after conception, when cells are really dividing, you know, very, very rapidly. And it's, you know, the incidence of early miscarriage is very, very high. I've forgotten what the outcome is, but I remember reading once that it's reckoned that only 50% of all fertilizations make it to babies.
Is it like 1 in 3 miscarriage before the woman?
There's miscarriages early on, are so common, and it'll be because, you know, there've been genetic mistakes in the copying process, the cell division process, and you know, they're very often just not viable. So yeah. We've then got the chemical mutagens, and benzoapyrene, not always got the A in the middle, which is why it's there in brackets. But yeah, benzopyrene, it's sometimes called in tobacco smoke, is, you know, one of the main mutagens, or we would call it, of course, a carcinogen. And it causes cancer by mutating the DNA. And there are other chemical effects on DNA that we can have that are not mutations. Because I was talking about epigenetics and, you know, the possibility for actually changing the nature of DNA and the histones as well. But, you know, I mean, given that tobacco smoke is reckoned to contain, well, it depends where you read, but I remember quoting on the GCSE something like 4,000 different chemicals, but I've seen bigger numbers, I've seen smaller numbers. A lot, anyway, and quite a number of them have the potential to cause mutations in DNA. And then we've got things like radiation, and we know that overexposure to X-rays can cause mutations that lead to cancer, UV light, you know, skin cancer. And these are all gene mutations, mutations being in the genes that control cell division. And you had 1 or 2 of those. I mean, you had the P53 has a role in that sort of area, and then also, I've forgotten which one it was. Was it the caspases? I can't remember which one of them it was that control. Anyway, I'm not going to go back to that. So we've got a variety of causes, and you know, the accidents, the biochemical accidents, are really, you know, what it is more than anything else. And we then have, down at the bottom here, different types of mutation. And these four have very, very different effects on the code, on the sequence of the DNA. Insertion is really just putting in an extra, well, it'd be a whole nucleotide, and if it happens on one side, then the enzymes will put in another one to match it on the other strand as well, so you end up with an inserted base in the code. Now the effect of that is going to be significant, because it doesn't just affect one amino acid. Well, it depends whereabouts on the strand it is. Deletion, obviously the reverse. All of a sudden we lose a nucleotide out of one strand, and that may result in one being cut out of the other side to make it match. Substitution, or sometimes what's called a point mutation, if it's a single base. Now, that's quite a common one, and to give you an example, cystic fibrosis is caused by point mutation. As is, I think, sickle cell anemia. So I'm going to come up with several other things that I can put on our list of possible subjects for the presentation. There's two for a start. Inversion, I think, is less common at the genetic level, but because I think, yeah, we don't find out about a lot of those because they're usually not survivable. Yeah. Mutations, yeah, well, if a mutation doesn't affect the individual who's got it, it's unlikely to affect their children either. It depends. You can be a carrier of a genetic condition, but usually if it's not going to show, that will mean it's a recessive version of that particular gene where you need 2 copies of it. So you would need them to have a child with somebody else who'd got the same mutation. So if it was a random one during the course of your life, it would be unlikely to affect your children. If, however, it produced a dominant effect, well, the chances are it's going to affect you anyway. But it does depend very much on where the mutation is and, you know, what its potential effects are. Right, if we have a look at the effects of some of these types of mutation then. Let me just park that out of the way. If we've got a little section of a gene, and you realise genes are probably thousands of bases long, a small section there that gives us an amino acid sequence of arginine, valine, serine. We've got 3 of the possible mutations, and the one that we haven't got is that inversion because it is very rare. But the substitution option, well, what can happen here? If we've got that sequence up at the top, what has happened? It says G converted to A, and where that has happened is at that point. Okay, so instead of GUU, it comes out with AUU, and the code is for a completely different amino acid. So the arginine and the serine are the same, but instead of valine, we've got isoleucine. Now, depending on what sort of amino acid that is, whether it's big, whether it's small, what sort of charge it's got on it, then it may or may not change the structure of the protein. So substitution might cause a big problem, it might cause a small problem, it might not cause any problem. If it had changed the base at the end of the triplet, it would actually be quite likely to produce one that is also a code for the same amino acid. We've then got the options of deletion and insertion. Well, what happens if you delete? Here they've deleted the G, all right, that G, and so you then shift everything up. So your triplets after that point completely change, don't they? So nothing is the same from there on. So we've actually then changed everything after the mutation has changed. So we now get phenylalanine and proline instead of valine, serine, and everything after that as well. Similarly with an insertion, of course, if we've added an A here, then that pushes everything on. So again, we get a completely different sequence after that. And these 2 types, I don't know whether this comes up on another slide, these 2 types of mutation are probably, the most serious that we'd be likely to encounter, and they cause what's known as a frame shift. frame shift. So it's as though you've got a reading frame, and it's actually moved that frame, and it's moved it on or it's pulled it back, so that you're not getting the same sequence at all after the point of the mutation. So the substitution one causes the least problems, is most survivable, I guess, would be the best description. It may still cause, debilitating conditions like cystic fibrosis, but this one is more survivable. Whereas, you change whole proteins, you may not then get an enzyme that keeps you alive. So, yeah, insertion and deletion are very significant. Okay, try some examples. Is the sheet which you've got following this, and I think we'll probably leave that for now because I think it's time for a break anyway, because I know the kettle's just boiled. So we'll take a break at this point, and yeah, break for all of us. I'll put it on pause and come. All right, let's have a look at a few more of the slides. I'm going to have to put that back to current slide. Right. So, looking at those examples, then you can get this idea that some effects are worse than others. So, insertion and deletion move the reading frame, so everything after that is a problem. It might be that if that was to happen in the very last triplet in the DNA for a protein, it might not matter. But if it happens in the middle, of course, it's then going to affect every amino acid after that. And it will depend what the sequence is. If you've got something, for example, like when we looked at the PowerPoint on the protein structure, we had fibroin, the protein for making silk, has a very simple sequence, and it goes alanine, glycine, alanine, glycine, alanine, glycine, alanine, glycine, and that's it. Obviously, you could disrupt that and perhaps, change part of that reading pattern, but if the rest of it was okay, then it wouldn't necessarily be a problem. So you won't necessarily just get the insertion of a single nucleotide. It could be 2, it could be 3, in which case that would be strange, wouldn't it? But, you sort of have to meet each individual mutation on its own merits, if you like, because, they're not always going to be equivalent. Okay. Now, the sorts of effects that we might see as a result of mutations, which we should now be able to identify from the examples that you've done, some of them are described as being silent, and a silent effect means that the polypeptide is still the same. So, that's where you are probably going to be producing exactly the same amino acid, just using a different triplet. You can have a neutral effect where the amino acid that's put into the polypeptide is similar chemically. If you go back and you look at those R groups, all right, they're quite diverse in themselves, but there are some that will be similar and may enable a protein to fold up in the same way, so you can replace one without it actually having a big effect. The frame shift ones, these are the ones that really cause problems. Anything after the mutation is going to be shifted backwards or forwards if you lose or gain a base. You can have what's known as missense, so that you still get an amino acid put into the protein and the protein continues to be made, but it doesn't work because it doesn't fold, it doesn't produce the shape that you want, so you just get this non-functioning protein produced. Or you can get nonsense, well, it's not really nonsense, but nonsense, where you get a stop codon suddenly inserted into it. So it doesn't allow the completion of the polypeptide. Because, of course, we've got, I think there are about other 3 codons, I think, or 3 triplets that code for stop. And, if that appears in the middle of your sequence, well, the protein is cut short. And therefore, you're not going to get a functional protein out of that. So, yeah, a lot of things can go wrong. But don't forget, that is why we are what we are. Evolution works on mutations. So, we've come a long way from being bacteria. This on your handouts is way too small to see. It may well be a bit small to see up here, but you know, if you you know, get onto Moodle and have a look at it, it's quite a good diagram. It's one of the Pearson ones. Pearson are really quite a good quality publisher. And you've got up at the top left, it's quite hard to trace what's actually going on, but top left, you've got your normal DNA producing a normal polypeptide, and the letters that go with them are actually underneath. So normal is up at the top. The next one down on the left is the one that's the silent mutation, where a change, where it read AAA and complementary UUU at the top giving phenylalanine, for the mutated one, this is a silent one where you've got a substitution where it now reads AAG and complementary UUC on the messenger RNA, still puts phenylalanine in. So that's your silent mutation where it doesn't have an impact on the protein that's produced at all. The one down at the bottom right is a missense one, where replacement of, I've got to try and check it up at the top, where it did read CGT on the DNA strand, it now reads CCT. So the mutated messenger RNA comes out GGA instead of GCA. So rather than having alanine as the third amino acid in that one, we've got glycine. So it's a different amino acid sequence. It is only slightly different, so it might not cause too many problems. Glycine being a very, very small amino acid, it might not cause, and actually it's been exchanged, alanine, of course, is the second smallest, glycine is the smallest, so that might not cause a problem. But of course, if it was some of those big ones with great big rings and things for their R groups, it could be really serious. Going over to the right-hand side, a nonsense mutation, and what we've got, the phenylalanine's produced okay, but instead of ATA in the normal DNA as the second triplet, we've got ATT that produces UAA in the messenger RNA, and that's a stop codon. So we just don't get anything else produced after that. So we lose, you know, from the original, the tyrosine, alanine, arginine sequence doesn't happen because the stop codon has just cut it short. A nonsense mutation we've got as an insertion is giving us that, where we've got an extra T inserted into the DNA, giving us an extra A in the messenger RNA. It is a frame shift one, moves everything on, and so everything after that is changed. So rather than get phenylalanine, then tyrosine, alanine, arginine, we get phenylalanine, but then we get isoleucine, cysteine, threonine, completely different amino acid sequence. Goodness knows what that would turn out to be. Similar sort of problem underneath that, of course, your deletion. So the frame shift deletion example, losing a T, you also would lose a U, and again, rather than getting tyrosine, alanine, arginine after the phenylalanine, this time you get leucine, histidine, valine. So, you know, it's a huge, huge difference. So that's actually a really good illustration of what happens, you know, easier than the ones that you have to write out for yourself probably, but yeah, your version of it, I think, unless you have incredibly good eyesight, because you haven't got the coloured version, but it's definitely sort of worth a look. I was yeah, quite impressed by that one. So you have got oh, that's interesting, yeah, the diagram on the notes there is actually one, I think it looks like that was the one I copied and used on the PowerPoint there. I'm going to go on with this, even though the other bits on here are not necessarily to do with genes, they're more chromosome based, but just to finish this off, 3 examples here. Cystic fibrosis caused by mutations. When it says mutations, it just means that there are quite a number of different types of mutation. There are different forms of cystic fibrosis, some of which are treated in different ways to others. The effects are different, and it's actually really a very interesting subject. I shouldn't have used this because it's got misspellings in it as well. main symptom is thick. No, it's got a mucus. Shouldn't have an O in it. Mucus, the noun, the thing, is just M-U-C-U-S. But if you describe a mucous membrane, you're using it as an adjective, then it's O-U-S. Right? The adjectives have the O-U-S, like famous describes something. So mucus, like that, is the stuff. But you are right, it is American spelling, because we have the next one, sickle cell anemia. We don't spell it like that. We have an A in there. So I really need to do my own version of this, don't I? This, when we get on to genetics, the idea of recessive for both those two will make more sense. But this is a mutation in the beta globin gene. Globin, if you remember, is the protein that forms the hemoglobin. You've got two alpha globins, two beta globins, and a mutation in the gene that produces the beta globin means that the red blood cells sickle when they are in low oxygen tensions, which of course is very inconvenient, in the tissues, and it means that they catch on each other, they block blood vessels, and so they interfere with normal circulation, as it says there. Huntington disease, or Huntington's, it's often known. That is at least spelt right. It's not Huntingdon, that's a place. Huntington, or Huntington's. But we do tend to refer to these conditions by just the person's name now. It's like Down syndrome, not Downs. And Huntington disease and Parkinson disease and Alzheimer disease. It's just it is a better description, but it doesn't matter particularly. This is a dominant condition, dominant version of the gene, and the mutations in this are due to some repeats, and I'm not sure if they're in the non-coding bit, but it affects the production of a protein called huntingtin, which is then affects the nervous system, and it's an interesting condition, but yeah, very unpleasant. The problem with this is that usually the symptoms don't come on until people have had children, and because it's dominant, of course, the chances of passing it on are very high. And there's a huge sort of ethical debate as to whether people should be tested for it if it's in their families, whether their children should be tested for it, because it is ultimately fatal. So, do you want to know that you're going to get it, or do you, want to know that your children have got it? But it's, one of those things that you could debate for ages. But all three of those are potential subjects for the presentation, looking at it perhaps from the genetics point of view. But when you do that, you then can't ignore what's come up in this unit, and you have to talk about the mutations and what effect it has on the proteins, you see. So yeah, I will add those to the list. Well, I've added two, and I will add Huntington's to it as well. Now, the other type of mutation that I mentioned then, because it's just worth keeping them all together, it seems a bit odd to do the genetic ones without the chromosomal ones. So we'll consider those while we're here, but you haven't got these in your handout because they don't fit for you into the biochemistry unit, but they're there on this PowerPoint. Some of these we're more familiar with than others. And yeah, so I've included a lot of images on here, I should warn you. And yeah, so yes, just a heads up. You can have the same sort of mutations in chromosomes as you can in genes, if you think about it, fairly obviously. These are not two different ones. I just liked the pretty colours of both of them. Couldn't decide between them, and so I put them both in. It's my PowerPoint. I'll do what I like. I think the bottom one I preferred, actually. It was a little bit clearer. It's also a good illustration of, don't overdo it with images on a PowerPoint. Choose the one, that shows things easiest. But you can, if you look across the top there, have deletion. You can lose a section of a chromosome and it becomes shorter. You can have duplication. When it's copied, a bit could get copied twice, like a bit of a hiccup in the copying process. You can have inversion, where chromosomes do all sorts of fancy things when they divide, and you can get loops in them. And if you get a loop which then breaks and is reinserted in the wrong way round, you can get inversion. You can get insertion, although to be honest, this one is probably, I think, what I would refer to as translocation, which is at the end there. But this one shows where a chunk of chromosome 4 has got inserted into chromosome 20. And you might think, well, would that cause any problems? You've still got the genetic information. Yeah, but it's not in the right place for being turned on and off in the right way. So, yes, it does matter. And at the end again there, you've got chromosome 4 and chromosome 20, and this time an exchange, really, I suppose, is what translocation is rather than an insertion. So a bit of chromosome 4 and a bit of chromosome 20 have changed places there. The other diagrams show exactly the same thing, but in a slightly different order, I think. So we've got a duplication first, then an inversion twisted around, a deletion where we've lost some, insertion where a chunk of one is stuck into another without an exchange, and then translocation where there is an exchange. So similar, very similar sort of process to the situation with genes. Now, theoretically, as long as a gene is still there, it should function. It's not always the case. Sometimes it is. And there are some quite sort of dramatic effects that you can get from chromosomal mutations. I'm not going into a lot of depth with these, but a fairly obvious one. You know what that condition is. That's Down syndrome. What causes Down syndrome? Too many chromosomes is the general understanding of Downs, and it's 3 copies of chromosome 21. Right? Now, if you think about it, then why is that going to be a problem then? Just because you've got an extra copy of all the genes on chromosome 21. Why does that result in this curious situation where individuals with Downs actually look more like each other than they look like their families? And it's a curious thing. It causes some very definite features, doesn't it? And the features that we're familiar with is the shape of the eyes, a single crease across the palm, a long tongue, very prone to heart disease, and a tendency to Alzheimer's as well. And the reason is because of the genes that are carried on chromosome 21. For example, Alzheimer's is largely still thought to be due to deposits of a starch-like protein, they call it beta amyloid, in the brain, which we do produce anyway, but deposits of this cause what they call plaques in the brain. And the gene for that is on chromosome 21. So if you've got 3 copies of it, we might describe it as being upregulated. You've got more of it, so it makes Alzheimer's more common. So that is Downs if you've got 3 copies of chromosome 21, but that's not the only form of Downs. There's actually 3 different types. Trisomy 21 is one of them. You then have, there's a translocation form of it, and I can't remember what's translocated where, but it involves chromosome 21. And then there's also mosaicism, where not every cell is affected. So the extra copy of chromosome 21 will have occurred after fertilization, and it'll only end up in some of the cells. That will give less or fewer disabilities. But yeah, the different forms of Downs. But we're most familiar with the trisomy 21, 3 copies of 21. All right, the chromosomes laid out in their pairs there, but that would be a Downs carrier type, we'd describe it as. The translocation's described there where it's a bit of chromosome 21 ends up on chromosome 14. So chromosome 21 isn't then functioning properly. And then the other one isn't illustrated there. This is a curious one, Turner syndrome, but not unusual at all. What are we missing? Well, we're either missing a Y or a second X. And looking at this individual, it suggests we're missing a second X, yeah? That's not quite the story. the development of human, I don't know, mammals generally, I suppose, in terms of what sex they are, what biological sex they are, what organs develop, is really controlled by the Y chromosome only. And if it's not there, the default is always female. And it's only a small part of the Y chromosome. There's one gene on the Y chromosome that's involved. All right, and that's it. And if that was to get translocated somewhere else and work, then individuals can look male regardless of what other chromosomes they've got. But this individual, yeah, female, because female is the default. The development of babies for the first about 8 weeks is totally sort of generic development. And at about 8 weeks, you then start to get the recognition of the Y chromosome and recognition of hormones in the body. And at that point, if there is a Y chromosome, the baby develops into a boy. And so nipples develop before that point, which is why men have nipples, all right? Because that's part of the generic development. Anyway, yeah, how you would describe this individual, they haven't got the full set of 46 chromosomes. You actually describe this particular condition as 22X0 or X0 or XO because there isn't a second sex chromosome there. So that'll be a loss of that chromosome when the cells are dividing to make eggs or sperm. There are impacts on development, tend to be very much smaller, it can affect mental development, and it's not an unusual condition. The reverse sort of condition for men, sort of, not reverse really, because you can't have a man without a Y chromosome, and you wouldn't have anything if you didn't have an X chromosome, because that's a huge chromosome, a lot of genes on there. But this one, Klinefelter syndrome, there's an extra chromosome, there's an extra X, but XXY, because there's a Y, you get the male development. But I really ought to trim that a little bit further, perhaps. But you can see it's not a typical male shape. There's a degree of sort of female hip development there, and that second X chromosome gives a degree of sort of what you might call feminization. Yeah, it's not yeah. So it's an extra one. Also has effects there. Now I can't remember what else I've got on here. Oh, this one. This is a translocation one, and it's a cancer, a blood cancer, leukemia, that's actually caused just by the translocation of a gene. The ABL gene is translocated from chromosome 9 to chromosome 22, and alongside the BCR gene, and there's an exchange of a bit in return. And it produces this little short chromosome with 2 named genes on there. It's known as the Philadelphia chromosome, and it leads to chronic myelogenous leukemia. I had a student with that condition one year, so not surprisingly, that was her presentation sorted. But yes, just moving a gene about. Weird, isn't it? What can happen? Oh, that was it. All right. So I can add quite a few probably onto the list over here. So in terms of ideas for presentations.